Do you have the homework problems in front of you?
Yes.
All right, well then, this would be a good time to review those problems. Let me take a look at question 1. Okay, so I'm looking here at question number 1. Now remember again, there's maybe 4 different types of acid-base problems that we've solved, summarized on the top half of that handout I've given you in the past. So refresh your memory for question 1 here. What type of problem is question 1?
Question 1 is a weak acid problem.
Okay, good. I gave you some problems like this in the previous homework assignment as well. And in that assignment, you got them right, but there were some problems where you were using a before change after table where you didn't really need one. All you needed was the Henderson-Hasselbalch equation. So I wanted to review that here. So I noticed that here you did not use the before change after table. You went straight to this mathematical equation. Now in this case, so this is the correct equation. You did this problem correctly, but this is the type of problem that I think we would have more understanding of if we did use the before change after table, because otherwise it's hard to see where this equation is coming from. Otherwise we're just doing this mechanically and we won't be able to adapt to any other types of problems. So for this type of problem, I think it would make good sense to use the before change after table and then see where this equation is coming from. In any case, you did end up with the correct answer here. Where did you get this number from, if you can see what I'm highlighting? What number does this represent?
The Ka.
So for this particular type of problem, do we need the Ka or the pKa?
The Ka.
So I just wanted to point out that this process that you did up here was correct, but unnecessary for this problem. What you did here is you figured out the pKa. And you did that correctly. This is the correct pKa. But you did not need the pKa here because you weren't using the Henderson-Hasselbalch equation. The Henderson-Hasselbalch equation used the pKa, but this equation does not. But you didn't make a mistake. You just did a step that turned out not to be necessary. Okay, so anyway, you did question 1. You did do question 1 correctly, but those were my comments on that. So let me take a look at question 2. What type of acid-base problem is question 2?
Question 2 is a weak acid and conjugate base problem.
Okay, that's right. So as we discussed last time, all you need for that is the Henderson-Hasselbalch equation. You don't need the before change after tables. You did this correctly. And in this problem, for this type of problem, do you need the Ka or the pKa?
For this problem, you need the pKa.
Why? Because the Henderson-Hasselbalch equation is about pKa, not Ka. Well, that was already given to you in the problem. The problem already told you the pKa, so there was no necessity to find the Ka. So that's good. So this is really all you need for question 2. These types of problems are maybe the simplest that we've seen so far. All you have to do is just plug into the Henderson-Hasselbalch equations. That's the exact right method for question 2. Let's take a look at question 2 is the type that I think you're most likely to see on the test. So let's take a look at number 3. Okay, so there's one problem with your answer here for number 3. You said the functional group was an amide and a carbonyl carbon. That's somewhat of a misunderstanding because the carbonyl carbon is part of the amide, not separate. So this group here is the amide. You could say that the amide consists of a carbonyl plus the nitrogen. So the carbonyl is not separate from the amide, it's part of the amide. The way you have it written here, it makes it seem like there's two separate functional groups, an amide and a carbonyl carbon. So here we could have just said the functional group is an amide. The carbonyl is just part of the amide. All right, but otherwise that's correct for number 3. So let's take a look at number 4. You can see you got that right here. When you drew the amide, you included the carbonyl. Okay, so you said that an amide is not basic. Why are amides not basic?
Because they have resonance.
That's a good start. Why does that additional resonance structure how does that additional resonance structure indicate that the amide is not basic?
Because the reason because of a double bond, like the resonance structure creates a double bond where there's a single covalent bond.
It does do that, but why would that mean that it wasn't basic? There isn't really any reason why that would mean it's not basic. So this is the right answer, but it seems to me that you're relying still a little too much on mechanical memorization, and we should try a little harder to understand the ideas we're talking about. So here's the amide. What was the most important factor in organic chemistry?
Charges.
So the key thing here is that the other resonance structure, the other resonance structure does introduce a double bond. That explains why the amide is rigid, because double bonds are rigid, and it explains why the amide is flat, because double bonds are flat. But it does not explain why the amide isn't basic. There's no rule that says the double bonds can't be basic. The part of the resonance structure that explains why this is not basic, there's two ways to look at it, but one way you can see it's not basic is that it already has a positive charge here. Does a positive charge make you more or less basic?
It makes you less basic.
Because what's the definition of a base?
Base is something that accepts protons and donates electrons.
But you wouldn't want to donate electrons if you have a positive charge. So one way you can see why this is a less basic resonance structure, why this resonance structure makes us less basic, is because it has a positive charge. Another way to see it is the reason why this picture is basic is because of the lone pair. But in this resonance structure, the nitrogen doesn't have a lone pair. So again, that would tend to make it less basic. Okay, so like I said, your answer here is correct. It's true that it's not basic, and it's true that it's based on the resonance, but it seems like we're still kind of relying a little too much on mechanical memorization and not understanding why the resonance structure made it less basic. The more we understand, the more straightforward the class will be. So the key is that the resonance structure has a positive charge, and things with positive charges are less basic. The double bond is important for it being rigid and planar, but it doesn't explain why it's basic. Okay, in any case, those basic answers are correct for number 4. Let's look at number 5. Okay, the proline is the part I especially wanted to review here because in the last assignment we had drawn proline incorrectly. If you remember, in the last assignment we added an extra nitrogen, but here you have it drawn correctly. The ring just joins up with the main chain nitrogens. That's a good picture of proline. What's interesting about histidine?
Histidine can take on two forms: positive charge and negative charge at physiological pH, or neutral charge and positive charge at physiological pH.
What's the major form?
The major form is the neutral form.
So that's what you drew here. But you should keep in mind that there's also 10% of the time it would be in the positive form. Okay, but this is a reasonable drawing because this is the 90% form here. Part B is very simple. You just list the amino acids from N-terminus to C-terminus. So does proline have a wide range of angles that it can rotate at, or a restricted range for Phi and C and Psi?
Restricted range.
Do you see how this ring makes it restricted?
Yeah.
Normally, there would be free rotation around a single bond. But when we put in this ring structure, there's much less freedom of rotation, especially for the phi angle. The phi angle is especially restricted here because it's locked into place here with the ring. That's an important point to see. That looks like a good answer for number 5. I think there's a good chance that on the exam they're going to ask you to draw a short peptide showing the correct charges. So this is why we need to have, one reason why we need to have all these amino acids memorized and know the ballpark pKa's. Okay, and why, you said peptide bonds are planar because of resonance. What is it about the resonance what is it about the other resonance structure that makes them planar?
There is no free rotation.
There is no free rotation, but that doesn't explain why they're planar.
They're is it because they don't have more than 4 attachments, so they're SP. They're not SP3.
That's pretty much that's on the right track. So the point is they are sp2 hybridized, and that makes them planar. It's really the same answer as for why they're rigid. So it looks like we've been having some trouble digesting those ideas here. But here's the resonance structure we're talking about. So because this resonance structure has, maybe I'll write these ideas down. The double bond makes it rigid and planar. Just like you remember from organic chemistry, alkenes are planar because of the double bond. That is related to the sp2 hybridization that you were mentioning. And the positive charge makes it not basic. So this resonance structure is interesting in 3 ways. Because it has a double bond, it makes the bond rigid and planar, because double bonds are rigid and planar. And because it has a positive charge, it makes the amide not basic, because things with positive charges are not basic. So the point is, none of those characteristics could be guessed from the normal resonance structure. This resonance structure looks like a single bond that's neutral, so it looks flexible and basic. But there's another resonance structure with a double bond and a positive charge, which makes the peptide bond rigid, planar, and not basic. So here's the way we can interpret this resonance structure to find the interesting characteristics of a peptide bond. Okay, do you have that down?
Yes.
Let's see, was there anything else I was going for here? Peptide bonds are rigid and planar. I guess that was what was it? Okay, good. So let's look at number 7. Okay, did you write these down from memory?
Yes.
Okay, good. So like I say, I think there's a real good chance that these will pay off for you on the exam. It's important to keep reviewing these every day so they'll be fresh in your mind when you take the exam. Remember that we've seen that in a free amino acid, the carboxy would be a 2. And in a free amino acid, the amino would be a 9. But you're usually going to be working with peptides. And in peptides, the pKa's are 3 and 8. So those are probably better numbers to use. Let's look at number 8. That is a good answer for number 8. So these are weak interactions. What's the name of the strong interaction? What's the alternative to these weak interactions that's stronger?
Ionic, covalent, and...
I think we're getting a little confused there. We've really only talked about one strong interaction. First of all, you mentioned ionic. But remember, ionic, I don't know if we talked about this, but ionic is just another name for electrostatic interactions.
Oh.
Ionic interactions is just another, because what is an electrostatic interaction?
Between a positive and negative charge.
Well, that's what we also call ionic. So in water, in physiological conditions, those ionic interactions are weak. So the strong interaction is just the covalent bonds. Strong interaction is just the covalent bonds. So this is the table we were looking at last time. The alternative to these weak interactions is regular covalent bonds, which are strong bonds. You have that down?
Yes.
All right. I just have to ask you since I can't see what you're writing. All right, so let's see. Okay, now there's a little bit more that I was looking for here for number 9. So for number 9, I asked you for an electrostatic interaction between 2 amino acids. Now, you kind of drew out this primary structure here, but what I want to go for here is to actually draw the whole structure out, kind of like you did down here. So let's shift to your screen. You were doing here glutamic acid and arginine. So try to draw both glutamic acid and arginine and show how, specifically by drawing out the structure, show the electrostatic interaction. Okay, now let me interrupt you. So the electrostatic interactions that we're focusing on are not usually between two adjacent amino acids. They're usually between two amino acids that are at different points in the strand. Mm-hmm. So rather than drawing these connected, we should draw them not connected, just as two separate amino acids that are on two separate strands. Because, yeah. So you can kind of draw that the strands kind of extend out. Mm-hmm.
Okay.
Your arginine, the way you've drawn it, I guess you should put a positive charge on the NH3.
Oh.
And because these are now separate from each other, you shouldn't have, you notice off to the side you have it written GLU-ARG. But that dash doesn't go there because that would be if they were sequential in primary structure. What does the GLU stand for?
Glutamine?
That's actually not correct.
Glutamate, sorry.
Yeah, glutamine is a different amino acid. Okay, let's see. What type of interaction is this?
Electrostatic.
Okay, so I just wanted to point out this is a little bit closer to what I was going for on this question. Remember that what we're trying to show is we're trying to show interactions that hold peptides in certain shapes. So this is maybe a better way to see what I was going for here. So notice here that we have a peptide strand. So notice that to save time, I haven't written all the amino acids here. This could be a whole bunch of amino acids. I'm just writing them as like a strand. What do you think that, so notice that this strand makes a U-turn. Instead of just being linear, it makes a U-turn. What do you think is what's the force that's holding it in this U-turn?
The electrostatic interaction.
This one right here, yes. All right, so this is a little bit what I was going for on number 9. Here's kind of what I meant by drawing an electrostatic interaction. This is an interesting electrostatic action between two amino acids because it shows how the peptide strand can be held in this shape. The shape of proteins is absolutely crucial because having the right shape is what allows the protein to fulfill the function that we're talking about. We want the protein to have a certain shape so we can know what function it's going to fulfill. Okay, so something like this is a little bit more what I'm going for showing the electrostatic, that type of electrostatic interaction. Okay, so then for the next part, I asked you to draw a hydrogen bond. I asked you to draw a hydrogen bond between an amino acid and water. You did that here. I also asked you to draw a hydrogen bond between two amino acids. Looks like you didn't get to that part. So let's try that on your screen as well. And let's try to make that picture maybe a little more like this picture. So let's try drawing a hydrogen bond between two amino acids in a peptide. A hydrogen bond between two amino acids in a peptide on your screen.
Between two amino acids and a peptide?
Yes. Kind of like what I did here. Do you see how this is a picture of an electrostatic interaction between two amino acids in a peptide, and it shows how the shape of the peptide is being supported by that. So I'd like you to try to draw a similar picture on your screen for a hydrogen bond between two amino acids. Now, one moment. Remember, what's the slogan for the atoms on the main chain is nitrogen, alpha carbon, carboxy carbon. So to be consistent with your earlier pattern, we have to start with the nitrogen. Yeah. Now it's on the right just because we made a U-turn.
Okay.
Okay. So which two atoms is the hydrogen bond between?
The tyrosine alcohol group and the
Now let's focus on the
tyrosine
particular atoms.
hydrogen on the hydrogen on the tyrosine and the oxygen on the serine.
Good.
Serine.
Good. And why are they capable of forming a hydrogen bond?
They are capable of hydrogen bond because of the delta positive charge on the hydrogen and the delta negative charge on the oxygen.
Okay, good. So I noticed that your peptide strand here made a kind of a U-turn rather than just being extended. What's holding it in that U-turn?
The hydrogen bond.
Yeah. Now, there could be other things too, but that's one of the things that's holding it. Okay, so that's a good sense of how hydrogen bonding can help us to maintain a particular type of shape. That's a very important topic. Let me see here. All right, otherwise those answers were correct for number 9. That's a correct answer for number 10. Okay. Let's see. So what are the 4 types of large biological molecules?
The 4 types of large biological molecules are proteins, lipids, nucleic acids, and carbohydrates.
Good. Now we're talking about proteins. Something I don't know if we've talked about much yet, but what are some of the functions of proteins? Can you think, what's one of the roles that proteins play in the body?
Proteins build muscle.
Let's see. I think you're thinking it like from a nutritional standpoint. So yeah, if you eat protein, that will help you to build muscle. But I want to look at more like at the cellular level. What actual functions do proteins play in the body? Can you think of any? What does the body use proteins for?
They use, I would assume, for energy.
That is certainly one function. That is one function. Of course, in a sense, that's not using them. That's just breaking them down. So what are the functions that proteins play? The most important.
They structure?
That's important, yeah. So there are structural proteins, absolutely. There are certainly structural proteins. The most important function I was going for is enzymes. Enzymes are proteins. This is the most important function that proteins play in the body. Do you know, what are enzymes?
Enzymes catalyze reactions.
That's right. What does that mean?
They speed up a reaction, or they lower the activation energy required for a reaction to occur.
Those are both good answers. So they speed up reactions without being used up in the reaction. Now you'll be studying enzymes more in just a couple of weeks. One key thing about enzymes is that they're highly specific. They only catalyze one particular type of reaction. Why are they so specific? Because the enzyme protein has folded into one particular shape. A shape that's only appropriate for catalyzing one specific type of reaction with one specific type of molecule. So I just wanted to mention the reason now that we're focusing so much on the structure and shape of proteins is because the reason proteins are so effective as enzymes is because they can take very highly defined and specific shapes. The ability to take on a wide range of highly specific shapes is what allows them to be so effective as enzymes. Now, proteins actually have many different roles in the body. Besides enzymes, another role would be as receptors. Proteins are receptors extending from cell membranes that allow messages to interact with the inside of a cell. Many hormones are proteins or peptides, so that's another important role. Antibodies, which play an important role in the immune system, are proteins, so that's another important role. And again, one key thing about antibodies is, if you remember from your biology class, there's a specific antibody for every different specific foreign antigen. Each antibody can only recognize one specific antigen because it has one specific shape. So antibodies can take a wide variety of shapes to specifically bind to a wide variety of antigens. So we need to, again, focus on what is it that allows proteins to maintain very specific shapes. Another type of protein is transport proteins. For example, some proteins transport things across cell membranes. There's also motor proteins that can provide contraction. There's probably tons of other things I'm not even thinking of. So proteins play many different roles in the body. It's good to be somewhat familiar with this list of roles that proteins play. Maybe the most important role is enzymes, but these other roles are important as well. And again, one key idea is that many of these roles are highly dependent on the protein taking a very specific shape. They're highly dependent on the exact primary, secondary, and tertiary and quaternary structure of the proteins. That's why we're focusing on that right now. Okay. So I wanted to give a little context to what we're talking about here. All right, so continuing, we were talking about weak interactions and strong interactions. Let's go back to primary structure. What's the name of the type of bond that holds together a primary structure? What's the name of the type of bond that connects amino acids in the primary structure?
Covalent bond?
It is a covalent bond. That's right. There's a special name for the covalent bond that connects two amino acids. What's the special name for the covalent bond that connects two amino acids?
A peptide bond?
That's right. But I did want to review, peptide bonds are covalent bonds, just like most of the bonds we talk about. Now, what are the types of weak interactions?
Hydrogen bonds, electrostatic interactions, and van der Waals
Good.
forces.
What is an electrostatic interaction?
Electrostatic interaction is the interaction or the attraction between a positively charged atom and a negatively charged atom.
Very good. What's another name for an electrostatic interaction?
Ionic bond.
That's right. What's a hydrogen bond?
Hydrogen bond is a weak attraction between a delta positive hydrogen and a delta negative oxygen.
Excellent. Let's see, that wasn't quite right. It doesn't have to be an oxygen. It could be between a delta positive hydrogen and any delta negative atom. Maybe the examples we've seen were with oxygens, but this is an attraction between a delta positive hydrogen and a delta negative atom. You actually could also, I guess, form a hydrogen bond between a delta positive hydrogen and an atom with a full negative charge, but this is what you'll usually see. Okay, so I think hopefully we're starting to get that down. So now we have to move on to van der Waals forces. Do you remember what van der Waals forces are? What are van der Waals forces? But between what types of atoms or molecules would you have van der Waals forces?
I don't remember.
Okay, so that's an important thing for us to discuss. That'll play an important role in this peptide chapter. Can these do these two strands have any electrostatic interactions between them?
No.
Do they have any hydrogen bonds between them?
No.
Why not?
Because there are no delta negative charges on either one or delta positive hydrogens on either one.
That's a good answer. So what's left to attract these? Well, what's left are van der Waals interactions. Where do the van der Waals interactions come from? Well, we do have a bunch of carbon-hydrogen bonds. What's the delta charge on this hydrogen? Delta positive, delta negative, or neutral?
Neutral.
Why neutral?
Because the electronegativity of carbon is similar to hydrogen, and there's no delta charge on either one.
Okay, that's a good answer. However, do you remember from chemistry and physics that electrons are always moving around at random? So what we said here is that the electrons should be pretty much halfway between them. The electrons will be halfway between them, so neither gets a positive or a negative charge. But remember, the electrons are not just stuck halfway between the two atoms in the bond. Electrons are constantly flowing around at random in any molecule. That's an idea from quantum physics. So even though on average the electrons will be halfway between these two atoms, because they're constantly sloshing around at random, at any instant, just by chance, the electrons might be closer to the hydrogen. And then in another instant, the electrons might be closer to the carbon. And then another instant, they might be closer to the hydrogen. On average, they'll be halfway in between. But any one instant, they might accidentally have vibrated to be closer to the hydrogen or closer to the carbon. What that means is that there can be temporary charges. There might be a temporary, say, a delta negative charge on this hydrogen. Now, this might only last for I don't know how long, a microsecond, a millisecond, I don't know how long it would last. This might only last for a microsecond, but just from the random sloshing around of electrons, there could be a temporary delta negative here. Now, when there's a temporary delta negative over here, that's going to repulse the electrons in this bond and create a temporary delta positive here and a delta negative here. This temporary delta negative would repulse the electrons in this bond and push them further away and create a temporary delta positive here. And then there can be an attraction between this delta positive and this delta negative. What's the name of this type of attraction? This is a van der Waals force. So, and notice how I'm using dots here.
You said a temporary positive or delta positive and temporary delta negative for the hydrogens?
That's correct, but I was trying to explain where those came from. Is that making any sense?
Can you explain that one more time?
Okay. So remember that because these have equal electronegativity, on average, the two electrons in the bond will be halfway between the carbon and the hydrogen. The hydrogen is not going to pull them closer, and the carbon's not going to pull them closer. But remember, the electrons are not just sitting there halfway in between. Do you remember the idea from physics and chemistry that actually the electron density is always moving around at random? The actual position of the electrons is random. Now, what we're really saying is that on average, they will be halfway in between the carbon and the hydrogen, but they're really just sloshing around at random. So occasionally, the electrons will slosh closer. You can almost kind of think of it like, I don't know, like water sloshing from side to side in a cup. You could think, so if we have here a cup, here's some water. Now, the surface of this water would basically be flat. It would basically be flat, but there's always some slight sloshing in the water back and forth. There's always some slight vibrations to the table that it's on. So at every particular second, there might be a little bit of an accumulation of water over here, or a little bit over here, or a little bit over here. Now, that's just temporary. Pretty soon, it dies down again. Overall, it's pretty flat surface at this top of the water, but there's always a little random sloshing. If it's on a pretty flat table, it's hard to see the random sloshing, but if you had a good enough microscope, you could see the random sloshing of the water. Well, similarly, even though these electrons are halfway between the carbon and the hydrogen on average, they do have a slight random sloshing motion, to put it a little crudely, between the carbon and the hydrogen. So at any one point in time, because of this random vibration or movement, the electrons might be a little bit closer to the hydrogen than to the carbon. And if the electrons were closer to the hydrogen than the carbon, that would create a little delta negative on the hydrogen and a delta positive on the carbon. Now that's only a temporary charge, just like these little sloshes here are only temporarily, because a millisecond later, the electrons will probably move back closer to the center or closer to the carbon. So these are delta negatives that are only temporary. They're only temporary because they're not created by electronegativity differences. If there was an electronegativity difference, there would be a permanent delta positive and delta negative. Because there's no electronegativity difference, the only reason that we had this delta negative is because of just the random movement of the electrons in the bond. But if there was a delta negative here, even if it was temporary, that delta negative would temporarily push away the electrons in this bond, creating a delta negative over here and a delta positive over here. And now that we have this delta negative and delta positive, there could be an attraction between them. Now that attraction would only be temporary. It would only be temporary. In another microsecond, it would disappear when the delta charges disappear. But that temporary interaction still is pulling these two molecules together. So that's what we mean by a van der Waals force. Now, a millisecond later, these might be neutral, and maybe a millisecond later, the van der Waals interaction will be over here, a temporary interaction over here. And then maybe a millisecond later, maybe now this one will be delta positive, and this one will be delta negative, just from the random sloshing around of the electrons. So the van der Waals forces come from these temporary delta charges that are a result of just the random movement of electrons, rather than the electronegativity differences. Okay, is that making any sense?
Yes.
Okay, so that's where the van der Waals forces are. Now, at any point in time, there would be lots of van der Waals forces, temporary van der Waals forces, at different points along these chains. So there might be a delta positive here and a delta negative. So that's what these van der Waals forces. So van der Waals forces are between temporary delta positives and delta negatives. So we can add that to our table. Van der Waals forces are between temporary delta positives and delta negatives. That doesn't mean the van der Waals force is temporary. The van der Waals force is not temporary because it consists of many, many interactions. Each of these individual interactions is temporary, but when it disappears, it'll probably be replaced by another van der Waals force on a slightly different part of the molecule. So overall, the van der Waals force, when you average together all those constantly fluctuating interactions, you get a fairly steady van der Waals force. Okay, I hope that makes some sense. In any case, this is the interaction between things that can't have electrostatic interactions or hydrogen bonds. If you can't form electrostatic interactions and you can't form hydrogen bonds, the only type of interaction that's left is van der Waals forces. Okay. Who's stronger, covalent bonds or electrostatic interactions?
Covalent bonds.
That's why we put this in the weak column. In the body, electrostatic interactions are weaker than covalent bonds. Who do you think is stronger, electrostatic interactions or hydrogen bonds?
Electrostatic interactions.
Can you think of any reason why the electrostatic interactions would be stronger?
Because they use actual charges instead of delta charges.
Good. Or they use full charges rather than delta charges. Delta just means a partial charge. Well, a full charge is going to have a bigger effect than a partial charge. Good. Who do you think is generally stronger, hydrogen bonds or van der Waals forces?
Hydrogen bonds.
Why?
Because hydrogen bonds aren't random.
Yeah, or more important,
Or
they're not temporary. These are permanent delta charges, where these are just constantly fluctuating temporary delta charges. Also, I would suppose these delta charges are usually smaller than the delta charges for hydrogen bonds. Okay, so these are all the different types. These are the key types of interaction that we need for your biochem course. This is a very important table to have down. Tell me when you have this table down.
Okay.
What's the name of, whoops. What's the name of this amino acid?
That is isoleucine.
Can this side chain, so how would this side chain interact with another side chain? What types of interactions is this side chain capable of forming?
It's only capable of forming van der Waals interaction.
Right. Can you give me an example of another amino acid that can only do van der Waals interactions?
Proline.
So what type of interaction would there be between these two side chains?
Van der Waals.
So that might be what's part of what's helping to hold this U-turn here in the peptide. Good. So this would probably not form van der Waals interactions with something that was polar, because something that's polar would prefer to form hydrogen bonds with something else that can form hydrogen bonds. These usually nonpolar things form interactions with other nonpolar things, because the polar things want to form interactions with other polar things. Now, usually, probably just one pair of side chains would be pretty weak van der Waals interactions. So you'd probably need a bunch of these side chains with van der Waals interactions in order to have a substantial amount of stabilizing force. I just drew one example of that. Okay, now there's one more important idea that's related to how proteins or compounds fold. So generally speaking, if you have something nonpolar, is it going to be hydrophobic or hydrophilic?
Hydrophobic.
Hydrophobic. So are hydrophobic elements going to try to, let's say you have a bunch of hydrophobic elements in water. Are hydrophobic elements going to spread out in water or clump together?
Spread out.
That turns out actually to be incorrect. That's not correct. Hydrophobic elements clump up in water. So let's see if we can understand why that is. I'll write that down. That's called the hydrophobic effect. what would water rather interact with? Does water prefer to interact with these nonpolar substances, or does water prefer to interact with other waters?
Other waters.
But notice these waters here are forced to interact with the nonpolar substance. The waters that are next to the nonpolar substance have to interact with it because there's no room. If you're right next to the nonpolar substance, there's no room here for any water kind of in this region. This part of the water is bordering on the nonpolar substance. There's no room to put a water here because the nonpolar substance is putting up that space, taking up that space. So the point I'm trying to make is along the surface of the nonpolar substance, water is prevented from interacting with any other waters. The surface of the nonpolar substance takes up space and prevents the water from interacting with other waters. So what do we want to do? We want to reduce the surface area of the nonpolar substance. If we reduce the surface area by having it clump up, notice what happens if they clump up. Do you see that when they clump up, there's now less surface area? When we put those two clumps together, now we lost half of the surface area here in the middle. So now there's less surface that the water has to border on. It takes fewer waters to surround the surface because there's less surface. It takes fewer waters to surround the surface because there's less surface. And therefore, there's more places for the water to interact with other waters. So the idea here is that hydrophobic things clump up, basically, so they have less surface bordering the water. It's kind of straightforward. We know that hydrophobic things don't like water. So do hydrophobic things want to have a large surface with water or a small surface?
Hold on. I wrote that they clump up so they don't get surrounded by water.
Now, that can't quite be right because, now, remember again, try to focus not so much on just copying what I'm saying, but understanding what I'm saying. What I'm trying to do is convey some concepts, and then you can write it down in your own words if you want. So the point is both of these are surrounded by water. You can see they're both surrounded by water, but the point is this has less surface area. Does that make sense? Do you see how that when two things join up, there's less surface area?
Mm-hmm.
Okay. Because there's less surface area, it takes less, so it's not, so they're both surrounded by water, but it takes fewer waters to surround the clumped object than the non-clumped. It takes fewer waters to surround this than this, because you can see here, the waters also have to be in this region in the middle. When they're not clumped up, we also need waters here in this on these surfaces. But those surfaces disappeared when it clumped up. So it's not that the water isn't surrounding it anymore. Water is surrounding both of them. But when it clumps up, there's less surface area to the hydrophobic substance. So there's less places where it's bordering on the water. Is that making any sense?
Yes.
Okay. If that's something that you understand, then the exact words you use to write down don't matter as long as you're writing down the right idea. So the idea here then is, so my question to you was, so does nature prefer for nonpolar substances to have, does nature prefer for hydrophobic substances to have a lot of surface bordering water or a small surface bordering water?
Small surface bordering water.
It's almost obvious from the name. It's hydrophobic, which means it acts like it's trying to get away from the water. Here we have a lot of the nonpolar substances in the interior, where it's not bordering the water, and a minimum on the exterior, where it's bordering the water. Now, technically, it's not that the nonpolar substance wants to get away from the water. It's more like the water wants to get away from the nonpolar substance. The water, it's not even, that's not quite right. Water wants to border as many other waters as possible. Water wants to be neighboring with many other waters. So the water wants to share minimum surface area with a nonpolar substance, so it can share more sites with other waters. All right, I hope I did an okay job describing that. In any case, the basic idea here is that the hydrophobic effect tells us that nonpolar things tend to clump together in water, and this is important because water is the key solvent in the body. Now, notice I didn't write this in the same table because this is technically not an attraction. We're not saying that the hydrophobic things, we're not saying that these two hydrophobic things attracted each other. They didn't attract each other. It's more like the water pushed them together. It's not so much that the hydrophobic things are attracting each other as that the water is pushing them towards each other so that they'll have less surface area. So that's a little bit of a subtle issue. But this is not a bonding. This is not a type of bonding or a type of force. But it's an effect of the fact that the water wants to be close to other waters, so the water tries to get away from hydrophobic things, and that tends to cause the hydrophobic things to clump together. Okay. In any case, these are the different ideas that we can use then to understand a lot of ideas in biochemistry, including protein structure. So, do hydrophobic things tend to spread out or clump together?
Clump together.
All right. I tried to do the best I could to explain why that is, but we can also just memorize that idea. All right, so let's see where we are now. All right, what are the two types of secondary structure?
Alpha, helix, and beta-pleated sheet.
So let's talk about the alpha helix. Is this the shape of an alpha helix? I was asking you, is this the shape of an alpha helix?
No.
Okay. So instead, an alpha helix. 1 second. Would look more like this. Now, this is a highly ordered shape. This is not something that would just naturally happen without a reason. If you have, say, a piece of string does not just automatically take on this highly ordered shape. So there must be some type of interaction that's holding this alpha helix in this shape. Now, I'm not sure, you might not remember from class what this is. Take a guess. What do you think is the type of interaction that's holding the alpha helix in this spiral shape?
Disulfide bonds.
Not a bad guess, but that turned out to be incorrect. It's not the disulfide bonds. So what it actually is hydrogen bonds. This is pretty likely to be tested. It's maintained by hydrogen bonds between main chain elements. You can tell me when you have that written down.
Okay.
So I'd like you to take a look at this structure and this structure. If these structures were close enough, could you form a hydrogen bond between them?
Yes.
Which two atoms would form the hydrogen bond?
The hydrogen and the oxygen. No. Actually, yes. Yes, it could.
Which two atoms would form the hydrogen bond?
The oxygen and the hydrogen.
And why can they form hydrogen bonds?
Because of their delta negative and delta positive charges.
So what would this group be? The hydrogen bond donor or the hydrogen bond acceptor?
Donor.
And would this be a hydrogen bond donor or acceptor?
Acceptor.
The one that's got the hydrogen is called the hydrogen bond donor. I don't know if I used that term before. So there could be a hydrogen bond between these two. But wait a second. They're not close to each other. How could they bond with each other if they're so far away? So it looks like these are so far away from each other that they couldn't form a hydrogen bond. Well, can you think of any way that these could form a hydrogen bond even though they're so far separate? And notice that this is 1, 2, 3, 4 amino acids away in the primary structure. How is it possible for a hydrogen bond to form between this oxygen and something that's 4 amino acids away in the primary structure? How could they be close enough?
If the molecule or the structure bends.
That's right. For what's one way it could bend? Well, it could coil like this. So for example, maybe this oxygen here is at this position. And maybe the nitrogen here is in this position. And because the spiral has coiled around, even though this nitrogen is 4 amino acids away, it's actually right in the same region of space right above this oxygen. What's the name of this type of interaction?
Hydrogen bonding.
Yeah, that's the hydrogen bond. So that's what I meant when I said hydrogen bonds are maintaining the alpha helix. These types of hydrogen bonds are maintaining the alpha helix, and it's exact this precise type. Now, are these between elements on the main chain or side chains?
These are elements on the main chain.
You can see I drew it right here. This is a main chain carbonyl and a main chain nitrogen. So the hydrogen bonds here are between carbonyls and the nitrogen then that's 4 amino acids further on. Notice here's an oxygen, and then 1, 2, 3, 4 amino acids further on is this hydrogen bond. And then this oxygen would form a hydrogen bond with this nitrogen, 1, 2, 3, 4 amino acids away. They look like they're too far away to hydrogen bond, but in the alpha helix, they'll coil close to each other. And then this oxygen will form a hydrogen bond with this nitrogen, 4 amino acids away. They'll be close enough because of the coiling of the bond. This oxygen will also form a hydrogen bond, but I can't draw it because now I'd be moving off of the page. It's hard to draw too many of these because it takes up too much space. But from this point on, every single carbonyl from this point on will be hydrogen bonded to the nitrogen that's 4 amino acids to its right in this structure. That's the pattern for alpha helixes. Remember that secondary structures are regular repeating structures. A secondary structure is a regular repeating structure.
So every single carbonyl on an amino acid alpha helix is hydrogen bonded
to the nitrogen 4 away. That's not quite right, because notice when you get to the end, for example, notice if this was actually the end of the peptide, If this was actually the end of the peptide, then this oxygen would have nothing to hydrogen bond with because he'd run out of nitrogens. And this one would have nothing to hydrogen bond with and nothing, this one. So almost all of the carbonyl oxygens will be hydrogen bonded, except the ones at the very end that don't have enough nitrogens to their right. That's probably a little bit more detail than your instructor is getting into in your course, though. Some courses go into that. But we don't need to go into too much detail for your course. So what we should say is all of the carbonyl oxygens are going to be hydrogen bonded to the nitrogen that's 4 amino acids to their right, except for the carbonyls at the very end that don't have a nitrogen, 4 amino acids to their right. And similarly, every main chain nitrogen is hydrogen bonded to the carbonyl that's 4 amino acids to its left, except for the 3 nitrogens that are on the far left that don't have carbonyls to their left. Like I say, you don't have to worry too much about the details. The point is that almost every carbonyl, so in the middle of the alpha helix, in the interior of the alpha helix, every carbonyl is hydrogen bonded to a nitrogen, and every nitrogen to a hydrogen on a nitrogen, and every hydrogen on the nitrogen is hydrogen bonded to a carbonyl. That is true in the interior. It's a regular repeating structure. Regular repeating structure. You can tell me when you're ready to keep going.
Okay.
All right. So it would be too confusing to draw all the hydrogen bonds. But for example, then there might be, so there would be tons of hydrogen bonds here. I'll just draw one more. Here's another one, but remember there would be all along the chain here, holding this structure in place. Okay. So what is it that maintains the shape of the alpha helix?
The hydrogen bond between the carbonyl carbon and the nitrogen hydrogen.
On the main chain or on the side chain?
On the main chain.
Okay, good. Let's see. So would you agree here, what was I trying to say, that if I start here and I start moving along, each amino acid will be slightly higher than the previous amino acid?
Each.
Do you see what I'm saying? If you started with your pencil here and you started tracing out the primary structure might be aspartic acid, glutamic acid, valine, proline, histidine, leucine, tyrosine. Each amino acid will be slightly higher than the previous amino acid, higher on the page.
Yes.
Okay. How much higher? Well, it turns out that in an alpha helix sheet, the rise, which is also called the translation, is 1.5 per residue. Do you know what this symbol stands for?
Angstroms.
Angstroms are a measure of what?
Is it length?
That's right. They're a measure of distance. Angstroms are a measure of distance. Good. Are they used for measuring short distances or long distances?
Very short distances.
So is it a long unit or a short unit?
Short.
Yeah. So 1 angstrom, it's got a little circle on top because it's named after a Swedish guy named Angstrom. And I guess the Swedish alphabet has this A with a circle on top of it. So this is equal to 10 to the negative 10 meters, which is 1/10 of a nanometer. So you can see it's pretty darn short. Nanometer is pretty darn short, and this is only 1/10 of a nanometer. So what I'm saying here is each amino acid on average in an alpha helix is 1.5 angstroms higher than the previous one. Each amino acid is 1.5 angstroms higher than the previous one. This is only for alpha helixes. This is only for alpha helixes. You probably need to memorize this number for your course. We'll see why in a moment. So there's a rise of 1.5 angstroms per residue. Remember, what's a residue?
Is that an amino acid main chain?
So a residue is just a name for one of the amino acids in a peptide. Residue just, so for example, how many residues do I have drawn here?
Seven.
Yeah. So if this was in an alpha helix, so what's the name of this amino acid?
Which one?
This one.
That is alanine.
And this one?
That is serine.
So in the alpha helix, the serine will be how much higher than the alanine?
0.1 or 1 angstrom. 1.5 angstroms over 7.
Say again?
1.5 over 7.
Not quite sure what you mean by the over 7.
Is it the rise is equal to 1.5 angstroms over the residue?
Yeah, I'm not sure what. Oh, okay. I think I might not have explained this clearly. The rise is 1.5 angstroms per residue.
Oh.
Like this. So there's no 7. I think you must have misinterpreted my handwriting there. So how much higher would the serine be than the alanine?
1.5 angstroms.
Right. What's the name of this residue?
Leucine or valine.
How much higher will the valine be than the alanine?
The valine will be 3 angstroms higher.
Because there's 1.5 angstroms per residue, but the valine is 2 residues further along. That's what I meant by per residue. This doesn't mean that the distance between any two things is 1.5. It's 1.5 per residue. So since the valine is 2 residues away, it would be 1.5 plus 1.5, or 3. Okay, good. That's an important idea. Another important idea is that in the alpha helix, there are 3.6 residues per turn. 3.6 residues per turn. Do you see what I mean by that? So for example, if you start here and then you move to this point here, how many amino acids are there between, say, this point and this point?
3.6.
And then how many amino acids are there between this point and this point?
3.6.
So how many amino acids are there between these 2 points?
7.2.
That's right, yes. So that's what I mean by residues per turn. This is, again, only for alpha helixes. You, again, probably need to memorize this number for your course. All right, so now I have a problem for you. What's the distance in angstroms between these 2 points? How much higher is, so this is 1 turn. What's the length of 1 complete turn? How would you figure out the length of 1 complete turn?
3.6 times 1.5.
That's right. How do you know that's right? Well, you could figure it out by looking at the units. What are our target units here? What units do we want our answer to be in?
Angstroms.
Because we want a distance. Well, we could start by saying that there are 3.6 amino acids on that turn. And then there's 1.5 angstroms between each pair of residues. So if we cancel, we would get 3.6 times 1.5. And actually, I guess the units now would come out as angstroms per turn. But those are even better units, because that's what we wanted to know. We wanted to know how many angstroms there are per turn. Do you have your calculator with you?
1 second. Yes.
Okay, so let's calculate how many angstroms are there per turn.
$5.4.
5.4 angstroms per turn. Now, do you see the difference between the 1.5 number and the 5.4? Here's 1.5 angstroms, here's 5.4 angstroms. If they're both angstroms, what's the difference between the 1.5 angstroms and the 5.4 angstroms?
Say that again.
Here I have a number 1.5 angstroms.
Mm-hmm.
Here I have a number 5.4 angstroms. They're both in angstroms, so what's the difference in how you interpret those two numbers?
The 5.4 represents how many residues? Or is 1.5 times the amount of residues?
That's not in very... try to put it into more common sense terms.
I don't understand what you're asking. Exactly.
I'm asking, what do these two numbers mean? What do they represent?
The distance, okay, so 1.5 angstroms is the distance between two amino acid residues, while 5.4 is the distance between 3.6 amino acid residues.
Right, or more practically, 5.4 angstroms is the distance of one turn in the helix. But what you said was right too. So this is the length of one turn, and this is the length between two amino acids. You might want to memorize this number as well, or if you don't memorize it, you need to be able to figure it out by multiplying these two numbers. So you could either memorize this 5.4 number, or you could figure it out by multiplying the other two numbers. Now remember, these are only for alpha helixes. Okay, let's try this problem. You can do this on your screen if you want, or you can just tell me how you would solve it.
Okay.
Okay, excellent. That's right. So you can use the units to see how to manipulate these numbers. Good. So you can see why I said this is a very typical type of problem for biochemistry exams. You can see why I said you have to have the 1.5 number memorized, or otherwise you wouldn't be able to solve this problem. Okay, so you can double check with your instructor about whether they expected to have that memorized, but most courses you would need to. Okay, that's good. Let's see. Okay, let's try this problem.
Okay.
What are the units on that answer?
Residues? There is no units.
Well, what does the number represent?
The amount of residues.
Yeah, so probably we should call it residues. That's right. Now notice this is only approximate. You can't actually have a fraction of a residue. If this is the complete peptide, you can't really have a fraction of a residue. The point is that it's probably pretty close to 86 or 87 residues. Close to 86 or 87 residues, because these numbers are all just approximate. Okay, so that's a good method for solving that. I just wanted to point out an alternative way to do this that I personally prefer. What were the target units on this problem?
Hello?
Yes, I asked you a question. I was asking what were the target units on this problem?
Residues?
What's the starting information? What information do we start with?
We start with the amount of angstrom, or the length in angstroms.
So I personally prefer to do this like a unit conversion. And then our conversion ratio here could be that there's 1.5 angstroms per residue. In the past, we wrote that as 1.5 angstroms over residues. But you can write it either way. And then you can see by canceling the units that what we need to do is 130 divided by 1.5. And that gives you the same answer that you got. All right, I personally prefer this method. I think it's more flexible, but there's nothing wrong with the way you did it. If you're more comfortable with the way you did it, that's fine. So you can stick with the method that you were using if that works better for you, but I think it's good to see that this is an alternative. Okay, I'll probably give you a few more questions like that in the homework. but to save time, let's move on. All right, so like I said, you might want to ask your instructor whether they expect you to have these numbers here memorized. Most classes you have to. All right. So your instructor in lecture seemed to emphasize one particular type of structure that has a lot of alpha helixes. So alpha helixes appear in many different proteins. Many different proteins have alpha helixes in them. Let's see. I want to talk about this now. Yeah. So your instructor particularly mentioned alpha-keratin. So I think there's a good chance that this will be on the exam. So alpha-keratin is the key structural component in hair, wool, nails like fingernails, claws, what else? Horns. Horns are really made out of similar material to hair. So alpha-keratin is a key structural component in all of these biological structures. It consists of 2 alpha helixes coiled around each other. So alpha-keratin is 2 alpha helixes coiled around each other. So we're not saying this happens in all proteins. This is what happens in this particular protein, alpha-keratin. Okay, you have that down?
Mm-hmm.
So for example, here's an alpha helix. Here's an alpha helix. Now, if you can imagine taking these 2 alpha helixes and coiling one of them around the other, you would get the structure of alpha-keratin. So alpha-keratin is a coiled coil. Because it's made from coiling 2 coils around each other. Let's see, so that would be hard for me to draw, but here's some pictures that your instructor gave you. So notice, here's the alpha-keratin. No, here's an alpha helix, and then notice here, when they take the 2 alpha helixes and they coil one around the other, this is the structure of the alpha-keratin.
She said, make sure not to write that it's alpha helix.
Actually, you're thinking of something else, because it is an alpha helix, isn't it?
Yeah. Oh, I'm at the entire structure. Like
no, I think you're thinking of collagen.
Yeah, collagen.
All right, collagen is a totally separate protein from the alpha-keratin. So collagen is a helix that's not an alpha helix, but we don't want to confuse those. So is keratin made of alpha helixes?
Yes.
Yes. Is collagen made of alpha helixes?
No.
No. Now, we haven't really started talking about them yet, but that's what you're remembering. So we'll talk about collagen next. But first of all, to finish off with alpha-keratin, alpha-keratin, as you can see from the picture, right? This is a picture of alpha-keratin, and you can see it really is made of 2 alpha helixes. Okay. notice that we could take this pair of coiled coils and then they can start intertwining those with other filaments as well. So this can add strength by taking the basic structure and then coiling that even more about other filaments. So this is showing the basic structure that alpha-keratin is made up out of. Okay, let's see. What type of molecule is alpha-keratin? What type of molecule is alpha-keratin?
Type of molecule. Is it a protein?
That's what I was going for. Good. It's a protein. Good. What's the structure of alpha-keratin?
The structure of alpha-keratin is alpha helix.
Try to be a little more specific. How is it different from any old protein with an alpha helix?
It's two alpha helix coiled around each other.
Yeah, two alpha helixes coiled around each other. Also, another difference is usually in most proteins, the alpha helix is just one part of the structure, whereas in the alpha-keratin, the entire structure consists of alpha helixes. The entire structure is alpha helixes in these pairs of coiled coils. Okay. Now, collagen is a little bit of a digression because collagen does not contain alpha helixes. So this is a little bit of a digression, but since your instructor talked about this, we can talk about this now. So collagen is the main structural component of skin, cartilage, bones, tendons, teeth. So you can see it's pretty important. Let's see anything I'm leaving out. Okay, I think these are the main things. So I think collagen is the most common protein in the body because you can see it plays such an important role in all of these very important structures. So what does collagen consist of? Collagen is a triple helix. Collagen consists of 3 peptide strands. Coiled around each other. In a triple helix. 3 peptide strands coiled around each other in a triple helix. Let's see, 1 second. All right. Now, those 3 peptide strands are not alpha helixes. The 3 peptide strands are not alpha helixes. So like I say, this is a little bit of a digression. Collagen does not contain alpha helixes, but since it is a type of helix, your instructor talked about this at the same time in class. So we may as well talk about it here. So what type of molecule is collagen?
Collagen is a protein molecule.
It's also a protein. That's right. And what's its structure?
It has three peptide strands coiled around each other.
Okay. Are they in alpha helixes?
Yes. No, it does not contain alpha helixes. So no.
Yeah. Remember, I think that was one of the main points your instructor was emphasizing in lecture. They're worried that you would get confused because they're talking about it at the same time as the alpha helix, but the collagen is not an alpha helix. It's basically a different type of helix. Collagen is basically a different type of helix. Now, the basic pattern of the strands well, let me show you. The textbook drew out the primary structure here for a strand. HYP is hydroxyproline. Here's the structure for hydroxyproline. How is hydroxyproline different from normal proline?
Has a hydroxy group on it?
Yeah, so you can see where the name came from. So the abbreviation is HYP. How many common amino acids are there?
20.
But there's also uncommon amino acids that you haven't memorized. You've only memorized the 20 common amino acids. Hydroxyproline would be a less common amino acid, but it's important in collagen. So here's the primary structure for collagen. Do you notice any patterns here? Take a look at what I wrote down here for the primary structure for collagen. Do you notice any interesting patterns?
Glycine, proline.
There's one very firm pattern. There's
Glycine
one.
occurs every 3 times.
That's right. So one thing that is crucial here is you need a glycine every 3 times. So there's going to be a glycine every 3 of these. The glycines are on the inside of the triple helix. So here's a picture of collagen.
Okay.
Do you see the triple helix?
Yes.
So this picture isn't great for me because I'm colorblind. But you can probably see there's a red strand and a green strand.
Yes.
So by using those different colors, they're trying to make it easier for you to see that there's 3 different strands here. So for me, the 2 of the strands kind of blur together. But if you can see the red and the green clearly, you should be able to clearly see there's 3 different strands. So this is what the triple helix looks like. Are these alpha helixes?
No.
Because it's collagen. Now, one thing to notice is, would you agree that on the inside of this triple helix, there's not going to be much room? Because they're all packed in close to each other?
Mm-hmm.
There's not much room on the inside of the triple helix because it's all being taken up by the helix. So that's why we need all the glycines. The glycines are all facing into the interior of this triple helix. Because what's the side chain for glycine?
The Side chain for glycine are just hydrogens.
Do you see how those would fit well in the interior because they don't take up much space? There's, if you saw from our picture here, every third amino acid was glycine because that's always the position that's on the interior. So this would be on the interior of the triple helix, exterior, exterior, interior of the triple helix, exterior, exterior, interior of the triple helix, exterior, exterior, interior of the triple helix. So that's why we need a glycine in every third position, because that has to go on the inside. That's the only thing that can fit on that tightly packed inside. All right, so one thing to notice is there's a glycine in every third position. The other thing is that you notice there's lots of prolines and hydroxyprolines.
Yes.
A lot of the other amino acids are prolines and hydroxyprolines. Would you agree those have to go on the outside because those are very large side chains? So they didn't show it here, but those would be pointing out from on the outside. All the prolines and the hydroxyproline side chains would be pointing out on the outside here because that's the only place that they can fit. Okay, so that's about all the time that we have. We'll have to finish up with this in a future session. But the basic thing to know here is that every third amino acid is glycine, and that many of the other amino acids are proline or hydroxyproline. What's hydroxyproline? Hydroxyproline is just proline with an extra hydroxy group on it.
I just want a second. I wanted to write down or draw that structure, and also mention no, the other structure, the hydroxyproline. The interior of the collagen is has hydrogen bonds.
Well, we haven't been talking about that, but you could ask what's holding the three things together. So the three helixes are held together by hydrogen bonds. Yes, that's not something we've really talked about yet, though. Maybe what you're confusing with is which amino acid is in the interior of the triple helix?
The glycine.
Why is that the logical one to put in the interior?
Because the hydrogens are small enough and they fit well
in the interior, because you can see how tightly packed the interior is there. The only thing that there's room for in the interior are the small glycines. The prolines are on the exterior so that their big rings can point outside. Now this picture doesn't show the rings, but you should imagine a big ring pointing to the outsides of this. So we hadn't talked about hydrogen bonds. We talked about the hydrogens in glycine that are on the interior. Okay, so anyway, that looks like that's our time. That's our time for today. So here's some things you should know. You should know what collagen is a component of. Your instructor emphasized it does not contain alpha helixes. So again, this is a little bit of a digression because we were talking about alpha helixes, but your instructor talked about collagen at the same time. So we're digressing to talk about that.